Welcome back, class. So at this point, we have talked about how T cells work, different types of T cells. We've talked about how B cells are activated, how they secrete antibodies, and how those antibodies work. We just have a few little bits and pieces of our immune system to throw in to wrap up our study of this chapter. So one of those are M cells. So microfold cells are cells that are kind of disguised here. Imagine that this is part of your intestinal tract, your respiratory tract, and this little cell in the middle, which is your M cell. It allows pathogens to come across. That pathogen thinks it's about to infect a cell, but instead it's going to find lymphocytes and antigen-presenting cells. So this is a way that your immune system is able to protect your digestive tract or respiratory tract. There are patches of M cells in your small intestine called Peyers Patches. And those Peyers Patches are lymphoid tissue in the small intestine. Here you can see a patch of it. This is a patch of lymphoid tissue in the small intestine, a patch of M cells that protect the small intestine from those bacteria in the large intestine. So if the large intestine biota start traveling up to where they shouldn't be getting into, those Peyers Patches will control that infection. Then there are NK cells. I already told you NK cells are my favorite because it's just fun to think of that, here I am, some boring professor person who has these killer deadly cells in my body. So NK cells are going to destroy virus-infected cells or tumor cells. What NK cells do is they look for cells that don't have MHC class 1. If you remember, MHC class 1 are things on a cell that signal to your immune system what's going on. So it'll be displaying good, healthy stuff if the cell is good and healthy, and it'll be displaying virus stuff if the cell is infected with the virus, or displaying tumor cells stuff if the cell is cancerous. If a cell does not have MHC class 1, it does not have any way to show what's going on inside of it. And so your immune system, your NK cells are going to say, I can't take the chance. I could let you grow, but if whatever is causing you to have lost your MHC probably is something dangerous. So your NK cells will release perforin, and you can see here's perforin being released. It targets the abnormal cell, and the abnormal cell will be broken down by apoptosis. This is nonspecific because it does not have to be stimulated by an antigen. It's the lack of anything antigenic that is going to trigger your NK cells to kill the abnormal cell. So how does a natural killer cell respond if the target cell does not have MHC class 1 molecules on its surface? Then a little bit more about what happens your second time. So antibody titer is the amount of antibody in the serum. It tells you, you have a lot of antibodies, you have a strong humoral response or a relatively weak. The first time that you are exposed to something, it's called your primary response. It is very slow antibody production. It takes really 7 to 10 days to start having any real measurable levels of antibodies. And then it peaks around 10 to 17 days. So a lot of infections, when you feel sick, you probably feel sick for about a week, maybe even 2 weeks before you start to feel better, because that's how long it's taking your primary response to mount a reaction. However, the second time, you get a secondary response or a memory response. So you already have memory cells in your body that can rapidly produce antibodies. So within 2 to 7 days, you'll start having an immune response. That's probably before you've even started having any signs or symptoms. You don't even recognize you were sick. And you also have a really greater response. So here you can see, here's the level of antibodies that you'd have for your first response, your primary response, and here's your secondary response. You might also notice class switching here. So here initially you have IgM, taking care of your immune response, and then that's going to shift to IgG. Here, even in a secondary response, you have IgM and IgG. So is a memory response primary or secondary? And then let's categorize our different types of adaptive immunity. So there's naturally acquired active immunity. Naturally acquired tells you got it somewhere natural, you got some infection, and it's active. You are making antibodies. So this is when a person has been infected and they have memory cells, they have active B cells and T cells. There's naturally acquired passive immunity. This could be something from a natural type of exposure, but it's passive because The antibodies are in something that's been transplanted or in colostrum. So the person with the antibodies isn't actively making any antibodies, but they have them there passively just from this transplant. But it is naturally acquired because it is natural to have those antibodies produced in response to the infection. Then there's artificially acquired active immunity. This would be in response to a vaccination or an immunization. When you've artificially acquired the response because vaccination is something scientific and not just nature giving it to you. But it's active because you are actively creating antibodies. And there's artificially acquired passive immunity. This would be when antibodies are injected. So it is artificial because it is injected, and it is passive because the antibodies have been injected. You aren't making antibodies in your body itself. Okay, so that's it for chapter 17. I think this is a pretty interesting topic, discussing our immune system and how we keep our bodies healthy. That is also the last lecture before your next exam. So at this point, you should be ready for your exam or spending the next few days getting ready for that exam. If you have any questions about it, please let me know, and I will see you later. Bye.